Hi, my name is Olivier Pardo, and I'm a team leader at Imperial College. And today I'm going to tell you about hnRNPA1 and how the phosphorylation of this protein regulates iris-mediated translation of particular mRNAs. So how did we get interested in hnRNPA1? Well, we're working for a while now on FGF2, fibroblast growth factor 2, and we had found that stimulation of lung cancer cells with this growth factor led to survival of the cells and drug resistance. We had also found that this was due to activation downstream of the MAP kinase pathway of a particular kinase called S kinase 2. This led to increased translation of antiapoptotic molecules such as BCL-XL and XIP in an iris-driven mechanism. So iris for internal ribosomal entry site. And the iris are 3D structures upstream of the AUG site of particular mRNAs that restricts then translation because it prevents scanning of ribosomes of these mRNAs. So these structures need to be unfolded before these proteins are efficiently sensitized. However, what we were missing is the mediator downstream of S6 kinase 2 that led to this increased translation. So we used two complementary approaches, one which was a tandem affinity purification for S6 kinase 2, and the other phosphoproteomic approach, both downstream of FGF2 stimulation, and we looked for the overlap. And indeed, 9 protein overlap between these two approaches, one of which was hnRNPA1. The reason why we got particularly interested in A1 was because there was already literature saying that hnRNPA1 was bound to mRNAs and regulated iris-mediated translation. So we first validated our finding, and indeed, if we stimulate cells with FGF2, we can see that hnRNPA1 is getting phosphorylated, associates with S6 kinase 2 in immunoprecipitation experiments, and S6 kinase 2 either in cells or in vitro is capable of directly phosphorylating hnRNPA1. This phosphorylation occurs on two sites at the extreme end terminal of the protein, serine 4 and serine 6. And the interesting thing is that only S6 kinase 2 in our hand is capable of doing this phosphorylation. Indeed, S6 kinase 1, which is 85% homologous to S6 kinase 2, or AKT, the prototypical AGC kinase, cannot do this phosphorylation. So what does this phosphorylation do? Well, it does regulate the ability of hnRNPA1 to regulate iris translation. So I told you that hnRNPA1 was known to be a regulator of iris-mediated translation, and indeed it's known as a repressor. So if you overexpress hnRNPA1 tagged either at the C or the N terminus, you can see that you repress translation of BCL-XL and XIP as compared to the affected control. On the other hand, phosphorylation changes everything. So if you look at the level of this protein downstream of FGF2 stimulation, we can see that in the vector alone, FGF2 increases the translation of both BCL-XL and XIP. Now, if we transfect the non-phosphorylatable version of A1, this does not take place anymore. There is no induction. On the other hand, if we transfect the phosphomimetic version of A1, the response is maxed out, whereby expression of this mutant increases the background level of BCL-XL and XIP, and now the further increase due to stimulation of FGF2 is not significant anymore. And this increase in translation is due to increased iris-mediated translation. And we know that because if we use a reporter assay driven downstream of the iris of BCL-XL, we can see that the phosphomimetic version of A1 increases translation downstream of this construct, but does not increase translation downstream of the construct that does not have an iris driving translation. So this is really specific to iris-mediated translation. So how does hnRNPA1 do all this? Well, it does it because it has the ability to shuttle in and out of the nucleus in response to FGF2 stimulation. And here we can see the cytoplasmic extract of cells stimulated with FGF2, and we can see first an export of hnRNPA1 out of the nucleus, followed by a re-import of the protein. And I'm going to show you right now how these changes modulate the translation of these mRNAs. So first, the export out of the nucleus. Well, it doesn't export on its own. It exports as part of the mRNA complex that exports mRNA out of the nucleus into the cytoplasm. And indeed, hnRNPA1 interacts with NXF1 and ALY, two molecules known to participate to the export of mRNAs from the nucleus. And if we silence NXF1, we can see that the export of hnRNPA1 out of the nucleus is cancelled. So the formation of this complex is necessary for A1 to exit the nucleus in response to FGF2. So as we described, this is an mRNA exporting complex, and therefore when hnRNPA1 comes out of the nucleus, it doesn't come alone, but it comes out with mRNAs such as that of BCL-XL and XIP. And we can see this here in this RNA-IP experiment where we pull down hnRNPA1 and qPCR for these mRNA, and in response to FGF2, we have an increase in export of both BCL-XL and XIP in the cytoplasm. This is dependent on the formation of the complex I just told you about, because if we silence NXF1, again, this export is cancelled. Also, it's dependent on the phosphorylation of A1, because if we express the non-phosphorylatable mutant, this export of mRNA does not take place anymore. So I told you that hnRNPA1 is known as a repressor of iris-mediated translation, but here I'm telling you that it increases the translation of these mRNA. So how does that happen? Well, it happens because the phosphorylation of A1 regulates differentially the affinity of A1 for mRNAs in the nucleus and the cytoplasm. So in the nucleus, the phosphonull version of A1 has got a decreased affinity for mRNA binding, while the phosphomimetic has got an increased affinity. On the other hand, once we go into the cytoplasm, this tendency is reversed, with the phosphomimetic version having a lower affinity for BCL-XL of mRNAs. So how does that switch happen? Well, it happens because 1433 interacts with hnRNPA1 and helps the release of mRNAs for translation. And this is because the phosphosites that I showed you on hnRNPA1 are in fact a 1433 consensus binding site. And indeed, our tandem affinity purification for A1 isolated 1433 theta, sigma, and epsilon as interacting with hnRNPA1. And this is validated here in a co-IP experiment where hnRNPA1 interacts with 1433 sigma and theta in response to FGF2 stimulation. Now, this interaction with 1433 is necessary for the re-import of hnRNPA1 from the cytoplasm into the nucleus. Here, we have the control cells where at late time point, like 30 minutes, hnRNPA1 goes out of the cytoplasm back in the nucleus. But if we silence 1433 sigma or theta, this does not take place anymore. And again, this is dependent on phosphorylation of A1, because if we express the non-phosphorylatable mutant of A1, A1 still goes out from the nucleus, but does not get re-imported into the nucleus. So how does that re-import into the nucleus occur? Well, it's downstream of simulation of A1. And this is because hnRNPA1 interacts with a whole simulation machinery when interacting with 1433 that plays the role of a scaffold. And we can see that indeed in late time points downstream of FGF2 stimulation, hnRNPA1 gets simulated. And this is dependent again on phosphorylation, because if we express the non-phosphorylatable mutant of A1, simulation does not occur and hnRNPA1 accumulates in the cytoplasm. 1433 interaction is required for this to happen, because if we silence 1433 sigma or theta, this simulation does not occur. We've isolated the site on hnRNPA1 that gets simulated, and this is K183. And as you can see here, if we mutate this site, hnRNPA1 does not get simulated anymore and accumulates in the cytoplasm. So what does that mean in terms of survival of the cells? Well, the phosphorylation of hnRNPA1 is required in order for FGF2 to mediate its pro-survival activity. And here you can see that if we treat this lung cancer cells with cisplatin, a chemotherapeutic drug, we increase the amount of cells going apoptotic. If we pre-treat the cells with FGF2, in the case of the control cells, this is totally inhibited and the cell survives cisplatin treatment. On the other hand, if we silence hnRNPA1, now you cannot rescue these cells from cell death anymore by FGF2 treatment. And phosphorylation is what really plays a role here, because if we overexpress a non-phosphorylatable mutant of A1, this survival again does not take place. It has consequences on the growth of tumor in vivo, because now if we express the wild type versus the non-phosphorylatable mutant of hnRNPA1 in lung cancer cell and inject these subcutaneously into mice, the non-phosphorylatable mutant containing cells can with a lot of difficulties develop into tumor as compared to the wild type containing A1. So we've got almost complete inhibition of tumor growth once A1 cannot be phosphorylated. So the data that I showed you today tells you that downstream of FGF2 treatment, what we achieve is cycling in and out of the nucleus of hnRNPA1, which exports mRNA into the cytoplasm to probe them for translation, and this release of the mRNA for translation corresponds to the association of A1 with 1433 simulation and re-import into the nucleus for another cycle. And the data that I showed you means that if we could inhibit either the phosphorylation of A1 or the association with mRNA, we could actually prevent the pro-survival effect of FGF2 and therefore increase the response to chemotherapeutic. And this is what we're doing now. We're developing inhibitors for hnRNPA1, and this is something that I hope to present you in coming years. Now, this research was almost entirely driven by my postdoc, Dr. Raj Roy, in my lab. This is a recent picture of my lab, but a whole lot of work is now going on in collaboration with Filippo Prisci in the University of Essex, where we have crystallized A1 and did a fragment screen to identify new inhibitors of this molecule. Thank you very much for your attention, and I hope you'll have some questions for me. Thank you.